Good evening, everyone. Welcome to the sixth talk in the elementary lecture series. In this webinar series, we invite eminent chemists from around the world to introduce the current frontiers of research in chemistry to undergraduate and school students. Today, we have with us Professor Padmanabhan Balaram, who will speak on chemistry, biology, and the unity of nature. Professor Balaram received his bachelor's degree in chemistry from Fergusson College, University of Pune, followed by a master's degree from the Indian Institute of Technology, Kanpur, and his PhD degree from Carnegie Mellon University. He did his postdoctoral studies at Harvard University with Nobel laureate R.B. Woodward. Following this, he joined IISc as a faculty member in the Molecular Biophysics Unit and became the institute's director in 2005. Currently, he is a faculty at NCBS Bangalore. He received the Shanti Swarup Bhatnagar Prize in 1986, the TWAS Prize in 1994, and the coveted Padma Bhushan in 2014. He is a fellow of the Indian Academy of Sciences, Bangalore, and the Academy of Sciences for the Developing World. So without wasting any more time, I would request Professor Balaram to begin his lecture. Professor Balaram, I think you're muted. You could unmute yourself and start. Yeah, you're audible now.
Yeah. Good evening. This evening I'm going to talk to you about chemistry, biology, and the unity of nature. What I want to do is really draw connections between the two disciplines and also try and think aloud about the manner in which these two disciplines help us to understand nature. On the very first slide that you will see on your screens, you will see pictured on your left Mendeleev, the man who gave us the periodic table, and on the right, Darwin, who gave us the ideas of natural selection and our understanding of biological evolution. In the middle, you will see the images, the many images which are now available on the internet of the coronavirus. And if there's one thing that the coronavirus has done, it's done many things. For example, it has taught us how to go online. It has taught us how to give online lectures. But at the same time, the coronavirus has awakened us to the enormous power of biology. The virus, in many ways, is an example of how powerful biology can be. And if I'm allowed to say this, I would say that this is one case where biology has trumped both politics and religion. Now my slide has got stuck. Why is that? Here we are. I hope you're able to see the second slide, but I will continue. This is a quote from Darwin's Origin of Species. And what Darwin said at that time was that there is grandeur in this view of life, with its several powers having been originally breathed by the Creator into a few forms or into one. And that while this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. These most beautiful forms include every biological species that you see around you. But it also includes many other things that you don't see, some of them wonderful and others not quite so wonderful, like the coronavirus, which we are dealing with today. Now, Darwin also thought about the origin of life, and that is something that I'm going to discuss. But in the 1860s, in the early 1860s, in a letter to a friend, he wrote that it is mere rubbish thinking at present of origin of life. One might as well think of the origin of matter. So in the 19th century, in the middle of the 19th century, neither the origin of matter nor the origin of life was very clear to anybody. A little bit later, he wrote another letter, and in this he said something remarkable. And when I read it, I was amazed. He said, it is often said that all the conditions for the first production of a living being are now present, which could ever have been present. And then he says, and this is the most important sentence if you are studying chemistry, is but if, and oh what a big if, we could conceive in some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity present, that a protein compound was chemically formed, ready to undergo still more complex changes. What is Darwin thinking about? This is one of the earliest writings in English where the word protein has been used. And here you can see the connection beginning to form in Darwin's mind, the importance of the chemicals of biochemistry forming before life itself could appear on Earth. But let's turn to biochemistry. When did biochemistry begin? The birth of biochemistry can be traced back to Edward Buchner's work approximately in 1896-97 on cell-free fermentation by yeast. And here what Buchner demonstrated was that if you took yeast extracts, the cells had been ground up and only the soluble soup was available, that soup was now able to convert sucrose into alcohol and therefore carry out the process that is called fermentation. Not everybody thought that this was a great discovery. Von Baer, who was Buchner's professor, really said that this will bring Buchner fame even though he has no chemical talent. I rather like this statement because somehow in chemistry there's been this great talk about chemical talent and what constitutes chemical talent. The 19th century chemists could have actually contributed greatly to biochemistry if they had been less focused on chemistry itself. But Buchner, who wasn't supposed to be a terribly good chemist, really is the founding father of biochemistry. But in the mid-19th century, Mendeleev began to write his periodic table. At the time he wrote it first, there were many, many gaps. Those gaps have now been filled up. And what I have pictured on this slide is really only a truncated version of the periodic table to fit into the slide. And I've now colored up the elements. All the elements which are green, hydrogen, carbon, nitrogen, oxygen, phosphorus, sulfur, are the elements of organic chemistry. They're also the elements of biochemistry. But there are many other elements which are there in light pink. I don't know what color appears on your screen. Sodium, magnesium, potassium, calcium, many of the transition metals. All of these are important for biochemistry. They're very important for life. I'm going to come back to the enzyme nitrogenase in a moment, but the enzyme nitrogenase contains both iron and molybdenum at its active site. In fact, today there are enzymes which contain the element tungsten in their active sites. So life uses many of the available elements on Earth. How did the elements themselves form? Your seminar series is entitled Elementary. And of course, elementary has two meanings. There is, of course, the famous meaning from Conan Doyle's Sherlock Holmes, where Holmes will always say after he solved a case, he will say, elementary, my dear Watson. That means it's very simple. But elements are the basic building blocks of molecules. And how do they form and where do they form? They form in the stars, of course. I want to draw your attention, and most of you are undergraduates, and therefore you should read. I want to draw your attention to this marvelous BBC series which appeared in the early 1970s, but which later appeared as a book, Jacob Brunovsky's The Ascent of Man. Brunovsky was a physicist, and he traced in this BBC series the ascent of man, and I'm going to come back to what the ascent of man really is. But Brunovsky wrote about matter. He said in all the stars there are going on processes which build up the atoms one by one into more complex structures. Matter itself evolves. The word comes from Darwinian biology, but it is the word that changed physics in my lifetime. In the few minutes that I had with a couple of students talking to them, they told me that they had not made up their minds which discipline they should take as a major. It matters little. because today biology, physics, chemistry, all of them are necessary in order to understand nature in all its glory. Carbon, for instance, is the most important element for organic chemistry. It is formed in a star when 3 helium nuclei collide at one spot within less than 1/1,000,000th of a millionth of a second. These are Brunovsky's words. And then Brunovsky is poetic. He says every carbon atom in every living creature has been formed by such a wildly improbable collision. Just think about it. 2 heliums colliding to give you an unstable beryllium, which then again collides with another helium, and then you get carbon-12. That's what is pictured, even if you look at Wikipedia. It's really marvelous. This is what nuclear physicists would study. This is what those who are interested in chemistry in the stars would study. But it is those carbon atoms which are formed and now are a part of the Earth. The carbon comes from the Earth itself after the Earth is formed. It is that carbon which is found in your bodies and in mine. I turn to biology. Here's a very famous statement by one of the 20th century's most articulate spokesman for Darwinian evolution, Dobzhansky, and he said, nothing in biology makes sense except in the light of evolution. This is the title of an article which he wrote in a journal called the American Biology Teacher. He was addressing biology teachers. And he says, does the evolutionary doctrine clash with religious faith? It does not. It is a blunder to mistake the Holy Scriptures for elementary textbooks of astronomy, geology, biology, and anthropology. Only if symbols are construed to mean what they are not intended to mean can there arise imaginary insoluble conflicts. This is a very important statement to think about because very many times, particularly Christian faith appears to conflict with biological evolution. Occasionally, even in India, there have been doubts raised in the recent past about the validity of the ideas that Darwin introduced on biological evolution. But today, the field of biology really rests on the pillars, or at least one of the pillars, that Darwin erected in the 19th century. What about chemistry? Chemistry has been described in this famous article by Arthur Kornberg. Arthur Kornberg was the biochemist who won the Nobel Prize for discovering DNA polymerase. And by discovering DNA polymerase, I believe he fired the first shot of the biological revolution in the 20th century. Kornberg wrote that chemistry is the lingua franca of the medical and biological sciences. Today, you cannot study medicine and you cannot study biology if you do not have a sound grounding in chemistry. Now, I suspect that since this lecture is being organized by the undergraduates, that many of you would have been born in the 21st century. And those of you who were born in the 20th century would have just been toddlers when the 21st century arrived. But 20 years ago, when the 21st century was about to arrive, there were lots of articles written looking back on the 20th century and also looking back on the 1,000 years, because we were also entering a new millennium. The journal Nature carried an essay every week, and I used to read them. They were wonderfully written essays in the year 2000 and the year 1999. But one essay remained in my mind, and I've picked it up and put it on this slide. This is an essay entitled Detonator of the Population Explosion and was written by a Canadian scientist, Václav Smil. And I quote only one sentence from his wonderful essay. He says, the world might be better off without Microsoft and CNN, and neither nuclear reactors nor space shuttles are critical to human well-being. But the world's population would not have grown from 1.6 billion to today's 6 billion without the Haber-Bosch process. It is now 7 billion. What is the Haber-Bosch process? It is something that you study in chemistry, and that's what I'm going to show you on the next slide. The Haber-Bosch process, which converts gaseous nitrogen and gaseous hydrogen under high pressure and temperature, using catalysts to produce ammonia is the first industrial synthesis of ammonia. It is this industrial synthesis of ammonia in the early part of the 20th century which really revolutionized all our lives afterwards. Why is that so? Because ammonia is the precursor for the industrial synthesis of urea, and urea is the first time that fertilizer was used in agriculture. And it is only after urea appeared and fertilizers were used that agricultural production grew up and famines receded, and there were very few famines after urea-based fertilizers began to be used globally in agriculture. But for those of you who are studying chemistry, or those of you who have studied chemistry and hate it today, the Haber-Bosch process is something you are undoubtedly likely to hate because it is a very boring reaction. But you should not be deceived by the way reactions look. It's important to see what is the product and how important is the product. In biology, ammonia is hugely important. And biological organisms, many of them have the ability to fix atmospheric nitrogen. And they do this remarkably at room temperature and in aqueous solution with the catalyst nitrogenase, which is an enzyme. So biology has evolved over the years to use all the elements that it is able to lay its hands on. But chemistry has other triumphs. In retrospect, some of these molecules are molecules which are hated. One of them is DDT. DDT, to give its full name, dichlorodiphenyl trichloroethane, is a remarkable molecule. It works to kill all kinds of insects, and as a result, it can be used both in medicine and in agriculture. It can be used in medicine to kill the malaria-causing mosquito. It can also be used in agriculture to kill the insects which are now damaging crop in the fields. Therefore, DDT took the world by storm after Paul Muller synthesized this in Switzerland. He was given the Nobel Prize in 1948. So we had one more chemical. Ammonia first influenced the world. The second simple chemical to influence the world was DDT. But DDT, of course, as it began to be sprayed extensively, in 1962, Rachel Carson wrote this book, Silent Spring. It's a classic. In that she says, we are rightly appalled by the genetic effects of radiation. She's talking about how appalled people were after Hiroshima and Nagasaki in 1945. And then she asked the question, how then can we be indifferent to the same effect in chemicals that we disseminate widely in our environment? This book is the start of the environmental movement. And today, when you hear people crying out loudly that we must protect our environment, you can think back on the simple book that Rachel Carson wrote in the early 1960s. So you can see that chemistry impacts life, chemistry impacts biology. But without chemistry, there can be no biology. New disciplines have come up. New disciplines have come up by a fusion of chemistry and biology. I have, for the last few years, been associated with the chemical ecology program at the National Center for Biological Sciences. And when I was asked to teach a course, the only qualification I had for teaching the course was that my chemistry was slightly better than the chemistry of the other people at the National Center for Biological Sciences. Apart from that, I knew very little. So I began to read. And I found this wonderful issue of the Proceedings of the National Academy of Sciences, in which Tom Eisner, one of the founding fathers of this field, had written this. New disciplines arise by a convergence of interest. Chemical ecology is the product of a partnership between biologists and natural products chemists, united by a shared vision and empowered by complementary skills. This is what I liked, because science is a collaborative enterprise. And if one can share a vision with other scientists, and if the collaborating scientists are empowered by complementary skills, the discipline can go far. What are the complementary skills that are needed here, and what is the vision? He says the vision stems from the realization that all organisms emit chemical signals and that all in their respective ways respond to the chemical emissions of others. This is true because except of us, no other biological organisms use language in all its complexity to communicate with other members of the species. By and large, they communicate via chemicals. So chemical communication in biology is central to biology. Even within your own bodies, one cell communicates with another via hormones and by other chemical transmitters. Your brain communicates with the rest of the body using chemicals. Therefore, chemistry is central to biology. Many other journals other than Nature also carried articles in the year 2000. This is an article by the famous molecular biologist Sidney Brenner. And his article began with a very provocative statement. He said, I once made the remark that two things disappeared in 1990. One was communism, the other was biochemistry, and that only one of these should be allowed to come back. I suspect both of them have come back or are still around. But he says in another part of his article, we do not have to resurrect biochemistry, and it will flourish because it provides the only experimental basis for causal understanding of biological mechanisms. Why did Brenner think that biochemistry was dead in 2000? He thought about biochemistry being dead because molecular biology had really swept away all the bridges to chemistry. Most people practicing molecular biology, and this was one of Kornberg's famous laments, was that all the bridges to chemistry had been simply washed away by the flood of molecular biological techniques. But underlying each one of these techniques was chemistry. Whether you're doing RT-PCR or anything else, there's chemistry underlying it. This chemistry had become so standardized that most people practicing these never even believed that there was any chemistry involved. But sometimes even simple chemistry can be useful. It can even take you to Nobel Prizes. Here's another one. This is from the Finnish chemist Arthur Kornberg, and he got his Nobel Prize in 1945. Much of the work that he did was during the war. And in Finland, in the cold countries of Scandinavia, after you've had your harvest, you need to preserve the fodder. How do you preserve the fodder? It'll just get slowly decompose. So he was looking for fodder preservation methods, and he realized that you must prevent harmful fermentations, Buchner's fermentations, which lead to the decomposition of proteins and the respiration of cells. He realized that all of these are stopped when the pH is lowered to 4, and he decided that the best way to do this was to add a mixture of sulfuric and hydrochloric acids. So the acids that are there in every chemistry laboratory have actually led to a Nobel Prize. He quietly adjusted pHs so that they dropped, and eventually he found he could preserve fodder right through the winter, the Scandinavian winter. And conservation of fodder in the form of silage and hay today is done in somewhat more sophisticated procedures, but not really very sophisticated. Today, you just want to add preservatives, common salt, sodium metabisulfite, phosphoric acid, and all you do is you limit the growth of undesirable organisms and increase the growth of lactic acid producing bacteria. This is exactly what you do also after you have taken, for example, antibiotics. You would like to have lactic acid producing bacteria grow in your stomach. You sometimes have a lot of curds for this purpose. That's biochemistry in action. So simple biochemistry can sometimes lead to profound advances in human well-being. Let's come back to modern biology. What are the three pillars on which modern biology rests? It might rest on more pillars, but I've chosen to rest it on three. These three pillars, and I'll name two of them, which were erected in the 19th century, are the ideas of evolution due to Darwin and the ideas of genetics due to Mendel. Both of these happened in the middle of the 19th century. The pillar which connects them together is chemistry. And here I have pictured on the chemistry pillar on the left in 1944, Oswald Avery's discovery of DNA as the pneumococcal transforming principle. Avery established that DNA was the molecule through which information was transferred from generation to generation. A few years later, Watson and Crick determined the structure of DNA, and everybody knows that story. What is interesting, of course, is that the Watson-Crick paper in Nature of 1953 does not carry a reference to Avery's paper itself. But what is the Watson-Crick double helix? It's a polymeric chains of deoxyribonucleic acid held together by hydrogen bonds between the nucleic acid bases, A bonds with T, G bonds with C. Hydrogen bonds are chemistry. The nucleic acid bases are chemistry. The sugar backbone is chemistry. The phosphate is chemistry. So chemistry is necessary for the DNA molecule to exist in this wonderful double helical structure, which allows it to be ripped apart and then used as a template for making new DNA. Mutations can be understood only if you understand the chemistry of DNA. DNA repair can be understood only if you understand the chemistry of DNA. So there's a lot of chemistry involved there. So biology, organisms and their behavior. The little picture that you see there is of the silk moth. The female silk moth, when it wants to mate with the male, signals to the male. It emits a molecule, the first pheromone, bombicol, very simple molecule, a fatty acid, slightly modified. This is volatile. And the male silk moth now senses this molecule with a receptor protein and then immediately moves towards the female silk moth. The entire silk industry depends on an understanding of the mating behavior of the silk moth. But that exquisite biology is controlled by chemistry. But I want to show you something before I move on. There are two dominant technologies of our times: information technology and biotechnology. You hear it all the time. And in the state of Karnataka, there is even a department which is called the Department of ITBT. The founding fathers of these fields I would put as Oswald Avery and Claude Shannon. Without DNA, there's no biotechnology, and without Claude Shannon and his mathematical theory of communication, there is no information theory. Both these are scientific advances which happened in the mid-1940s and the early 1950s. And interestingly, there's a connection. An algebra for theoretical genetics is the title of the PhD dissertation submitted by Claude Shannon to MIT in 1940. Today, of course, as the genomics revolution moves on, nothing in genomics is possible without the enormous advances that have taken place in computer science over the last 50 or 60 years. We've seen Mendeleev's periodic table that hangs in every chemistry laboratory and nobody looks at it. The metabolic pathway chart hangs in every biochemistry laboratory, and I suspect nobody looks at it because it's just too complicated. If you go very close to it, you might be able to see a few chemical structures. But today you have interactive maps on the internet with which you can play. It's remarkable. It's reactions and reactions and reactions, all of them coupled to one another, which allow the cell to work in this wonderful way that it does. If our cells don't work so precisely, we are not going to be around for very long. And therefore, one must look with huge respect on metabolic biochemistry. Metabolic biochemistry, which is probably the most unglamorous area of biochemistry for teaching, and I'm sure when you hear about it in your undergraduate classes, you wait for the sections on metabolism to get over so that you can now look at sections on transcription and transcription regulation and so on. But remember that biochemical metabolism is making a major comeback in the 21st century. There is this wonderful review article which I will draw your attention. It is in Chemical Reviews in 2018, but by one of the most foremost biochemists in the world. And this is asking the question, what is the logic of metabolism? It is so complex. How did it evolve? nature must have logic in order to put all of this together. What is that logic? And they now come down to a very interesting statement that there are 8 central molecules for all of biochemistry. I'm not going to tell you what those 8 central molecules are, but on my slide there's a reference. Those of you who are interested can go and look at those 8 central molecules. All of them will be very simple molecules. But I want to come back and show you something else, because I am really trying to draw connections between chemistry and biology. And I am sometimes distressed by the fact that people think that chemistry, biology, and physics can sometimes be thought of as completely distinct subjects. All these disciplines need one another in one way or the other to understand something. Look at the red chili. All of you like it, I'm sure. And the molecule which gives it its very spicy taste is capsaicin, the vanilloid structure that you see on the right side of my slide. You can ask the question, how does the red chili make this molecule? And on your left is the biochemical pathway, the biosynthesis by which this molecule is made. A set of chemical structures being transformed into one another. And slowly you have 2 lines, one beginning with the amino acid phenylalanine, another beginning with the amino acid valine. They are the starting materials. Eventually you go through biosynthetic pathways and you come up to 2 other molecules. These 2 molecules are now combined with one another to give you capsaicin. So it's a very complex synthesis, biosynthesis. On each arrow there is a name. Phenyl ammonia alanine ammonia lyase is the first on the first arrow. That's an enzyme. That's the catalyst for the first conversion of phenylalanine to cinnamic acid. So everywhere you need biological catalysts, which are enzymes. Sometimes those biological catalysts may or may not be known even today. But as you go down, you realize one thing, that if you were in an organic chemistry department, you would be taught this, and the phenylalanine ammonia lyase would just be a name. But if you are in a biochemistry department, you would concentrate on the enzyme, and all phenylalanine and cinnamic acid and coumaric acid would now just be names. So sometimes it depends on your perspective. Unfortunately, nature does not draw any distinctions between chemistry, biochemistry, and physics. It uses all of them as and when it wants. And this is something that you must actually keep in mind. But how are those catalysts produced? To produce every enzyme, which is a protein, you need a gene. So you can now imagine how much of genetic information you need in order to produce capsaicin. A simple organic molecule requires a huge amount of genetic information. And I want now to really tell you a story. I've now told you something about chemistry, something about biology, something about their importance for one another. Now, in the beginning of this year, in the magazine New Yorker, a very interesting article appeared called "Does Hurting Make Us Human?" And they found this lady, a lady in Scotland, Joanne Cameron, who, because of a combination of genetic quirks, her emotional range was very limited. But what is amazing is her negative emotional range was, so she couldn't feel pain and she didn't have any capacity for negative emotions at all. So when she went in for some surgery which was very painful, she told the anesthetist that she doesn't want anesthesia. He, of course, gave her anesthesia, but when she came out of anesthesia, she told the nurse that she doesn't need a painkiller. So the nurse didn't give her a painkiller and recorded this on her chart. When the anesthetist came back the next day to check on her, he found this remarkable observation. So they then took her up for investigation and found that she had a genetic problem, and the genetic problem was a biochemical problem. There was this wonderful molecule which I'm going to tell you about, Anandamite. This is there in your brain, and look at its name. It's an Indian name, Anandamite. And what happens to anandamide in the brain is that it is broken down by an enzyme, a fatty acid amide hydrolase. It is broken down to arachidonic acid and ethanolamine, the very molecules from which it is biosynthesized in the reverse reaction. But if you don't break anandamide down, it accumulates in your brain and it makes you feel very good. So why does anandamide make you feel good? because anandamide actually acts on the same receptors where the active constituent of marijuana, that is tetrahydrocannabinol, acts. But tetrahydrocannabinol and marijuana was known, hashish was known for a long time, and in the 1960s, an Israeli chemist whom I'm going to show you, Raphael Meshulam, he discovered the structure, he established the structure of tetrahydrocannabinol. This is the active constituent of marijuana. He now wanted to know to what receptor it bound, and if there was a receptor for this, obviously the receptor was not there for binding tetrahydrocannabinol, it must be there for binding another molecule. So he discovered the structure in tetrahydrocannabinol in 1964. In 1992, he published a paper he'd extracted a molecule from pig brain, anandamide. And this is simply from the paper itself. This appeared in Science, and I've taken this paper, the PowerPoint now, because I'm using a newer computer than the one on which I made this slide. Looks slightly different, probably. The term anandamide was coined from the Sanskrit word ananda, which means bliss, and from the chemical nature of the compound. On the left is the kind of assay that you would do in a biology lab, a immunoassay, a radio-labeled cannabinol being displaced from rat brain synaptosomal membranes by cold ligand. On the right is what you would do to establish a chemical structure, take a mass spectrum, and try to find out what it is. So both chemistry and biology in Meshulam's work, wonderful work. He was asked in an interview, why didn't you use a Hebrew name? He said, we looked for a Hebrew name, but as you may well be aware, Jews are not very happy. We have a lot of words for being down and so on, but not so many for words of extreme joy. So you can see that our ancestors have been slightly better off than the Jews of yore, and they have coined words for extreme bliss. But coming back to biosynthesis, substrates must be converted to products. They need enzymes, and enzymes need genes. So for any multi-step biosynthetic pathway, you need gene clusters, you need operons, and you need modular assembly of all these genes. But the very fact that it's very difficult to make a small molecule was realized by Francis Crick. I used this slide simply for those of you who are enamored by molecular biology. There's no other way to turn your attention to chemistry than to quote from one of the high priests of molecular biology. And what Crick said was this: the main function of proteins is to act as enzymes. And then he said, it is at first sight paradoxical that it is probably easier for an organism to produce a new protein than to produce a new small molecule. Looks paradoxical. And then he said, there seems little point in genes doing anything else but protein synthesis. He said this in 1958. But molecular biology has moved since then, and by 2000 the Human Genome Project had been completed, or at least politically completed. Today we have as many as 789 genomes some time ago, and these genomes can be compared, and you can draw trees which link all the eukaryotic organisms, and you can see where the humans are on a branch with the chimpanzee, not too far away from the rat and the mouse. And I rather like this tree because it puts pigs, cattle, sheep, all of them in one branch, and all these branches are not too far from human beings and chimpanzees. This is what I meant really by the unity of nature in their genetic composition and in their biochemistry itself. Today, there are a very large number of genomes being determined across all the way from archaea to bacteria up to eukaryotes, viruses, and so on. And there is a site on the internet where you will get all this data, and you must go and look at it. This tells you what modern computer science can do to allow you to cope with the flood of data. But I'll come back towards, as I reach more or less the second half of my talk, to the timetable of evolution. This is taken from a recent review a couple of years ago in Science Advances, where you can see the Earth has been around for about 567 million years, 5.67 billion years. So at the beginning, there was only the Earth. It was hot. It was, nobody knows exactly what it was, but It was pretty terrible. And that is the period which is called the Hedian Period by those people who are interested in the origins of early Earth and all the ages which came afterwards. But by the time we're about 4 billion years ago, we begin to see a geological age now being described in biological terms, the Archean. And by this time, origin of life is probably around somewhere between 4.5 billion years ago and all the way up to about 2.5 billion years ago. And biological carbon and sulfur cycles, which are chemical cycles, are beginning now. And so by the time you reach 2.5 billion years ago, you have archaea now inhabiting the Earth, and archaea live largely, most of them today, in an anaerobic atmosphere. And therefore they live in an atmosphere without oxygen because early Earth had no oxygen. So it took much later for photosynthesis to appear and oxygenate the atmosphere. This is what these Earth scientists would call the grand oxygenation event. And that happened somewhere between about 2.2 billion years ago when oxygenic photosynthesis began and the atmosphere began to get rich in oxygen. Once that happened, multicellularity began and the origins of animals also can be traced back. What is interesting here, of course, in the Cambrian period, there was a lot of animal diversification, although recent papers suggest that some of this might have happened even before. This came somewhat later. And today, when we look at nature around us, we recognize plants, we recognize animals, we recognize everything that we see. But a lot of what we don't see, microbiology, bacteria, archaebacteria, many other kind of very simple eukaryotic organisms, they are there and they are there in abundance on Earth. Primates and the hominoids came very, very much later. So the dinosaurs disappeared around 66 million years. And those of you who are undergraduates, I hope, still have the kind of imagination to see Jurassic Park and wonder what the world would have looked like when the dinosaurs were still around. So the timetable of evolution really depends on all these factors, the grand oxygenation events. There are other oxygenation events which have taken place later. And if you look now at at contemporary biochemistry, you will find oxygen is most important for respiration. There's a famous experiment, the chemical evolution experiment done by Stanley Miller in the late 1940s, early 1950s, published in 1953, where he took ammonia, methane, hydrogen, and steam in a glass bulb, and then had a spark discharge pass through it, and then collected in a flask at the bottom. He collected what I now believe is a brownish black mass preserved at the University of Chicago. And then he wrote a paper on the next slide. The paper, which appeared in the same year in Science as the Watson-Crick paper appeared in Nature, shows a paper chromatogram where he discovered the amino acids. So he had produced an amino acids from simple gases. Interestingly, the New York Times picked up this paper. They didn't pick up the Watson-Crick paper. They put it on the front page of the New York Times and called it the Origins of Life Experiment. This is the Origins of Biochemistry experiment. Many years later, over 50 years later, one of Miller's former students went back, now quite a mature scientist. He went back and took the same sample from the University of Chicago and subjected it to the latest techniques of analytical chemistry, liquid chromatography and mass spectrometry, and he discovered so many more molecules. So what we see really depends on the analytical techniques that we have available. After the original cells were formed, they diversified very quickly. Now, of course, you might ask, what was the last universal common ancestor? What was the common cell, one unicellular organism from which everything else diversified in this tree of life that you will see? In this tree of life, which you should look at somewhat carefully, you will find human beings occupy a very small place. So when we think we are very important, biology will bring us back to the ground state. We realize that there are many other things in biology. But in the current literature, and this is a paper in Nature in 2020, progress is being made in trying to understand what the ancestor of eukaryotic cells might really have looked like. And this is really very exciting. What do we know about prebiotic chemistry? Hydrogen cyanide, if you take, you can make all these molecules. Those of you who are chemists can see simple reactions. All we are using here is hydrogen cyanide and a little bit of light. If you look down below, you find that if you add a little bit of water, if you add a little bit of isocyanate, you can make many more molecules. You can get the nucleic acid bases. This is the remarkable kind of prebiotic chemistry that one can think about, which hydrogen cyanide as the central precursor. But remember, hydrogen cyanide here on this slide is a precursor for life. Hydrogen cyanide was what was used to kill 6 to 7 million Jewish prisoners by Hitler during the Second World War in the gas chambers. He generated hydrogen cyanide using the capsule which was then called Zyklon B. The chemistry for this was again the chemistry which was done by Fritz Haber. Today you can look outwards, you can look into outer space, you can look at comets, and you can analyze the molecules or the chemistry that is there in comets. And the Hale-Bopp comet, for example, you will find the simple molecules that are present. But I have put arrows only for two molecules, formaldehyde and hydrogen cyanide, because I'm going to show you what happens on the next slide. If you have formaldehyde and hydrogen cyanide, you can make many more molecules. And this paper, which appeared in Nature Chemistry in 2015, tells you that if you have hydrogen cyanide, hydrogen sulfide, a little bit of copper, light, you can make all of this. So you can see biochemical complexity begin to appear before your eyes without any biology whatsoever. Is this the precursor of biological evolution? Definitely so. But there must be a lot more chemistry to be discovered before we build a bridge between chemical evolution and biological evolution. In this book, which I've been reading for some time now, Nick Lane asked the question, why is life the way it is? Bacteria evolved into complex life just once in 4 billion years of life on Earth, and all complex life shares many strange properties, from sex to aging and death. If life evolved on other planets, would it be the same or completely different? I suspect it would really be the same, and that's the conclusion to which one must come. If one look at the evolution of the elements in the stars, if you look at the evolution of the molecules on Earth, you would imagine that there were Earth-like planets, maybe the same kind of biochemistry would have evolved there. In the last part of my talk, and now I'm going to move very quickly, I'm just going to show you something that I've taken from the internet. 200,000 years ago, what would the world have looked like? It would have looked like this. There would have been hardly any human beings, the precursors of human beings, the few hominoids coming in East Africa, where you can see that little shade of grey, where I will put my pointer. Just before I began this talk, yesterday in my rehearsal and today, one of the undergraduates taught me that I can use a laser pointer even in these presentations, so I must try. That's where the pointer is. 32,000 BC, we've gone 170,000 years, and the grey has spread now. And it's spread all over. It hasn't still spread to the Americas, you can see. The time we come to the beginning of the current era, you can see it's come. All the civilizations that we talk about, India, China, the Middle East, Persia, the Mediterranean civilizations, all of them have come. Nothing much in North America, nothing much in Australia, nothing much in Africa or South America as yet. Now, of course, if all these civilizations have come, we can go back and look at fossils. We can extract DNA from these fossils and ask, how did this kind of migration happen? I draw your attention to a book by Tony Joseph called The Early Indians, which came out some time ago. And really it turns out that ancient DNA may rewrite the prehistory of all our countries, India among them. In the Andaman and Nicobar Islands, you see this picture of Indira Gandhi many years ago, sort of with the natives who were there in the Andaman Islands. But there are one or two islands still where there are hunter-gatherers still present, largely untouched tribes. The ancient DNA boom is very recent. It is only in 2014, the year I retired from the Indian Institute of Science, that you will see this. And then after that, there has been this exponential rise. Today, when I made this slide in 2018, there are 7,316 samples. There are probably more. But ancient DNA must come from fossils. So many branches of earth sciences now integrate with biology here. Biology and history also integrate, and this brings you really to the final part of my talk. You must read this book if you have not read it. Sapiens by the Israeli historian Yuval Harari. He writes that the immense diversity of imagined realities that sapiens invented and the resulting diversity of behavior patterns that are the main components of what we call cultures. Once cultures appeared, they never cease to change and develop, and these unstoppable alterations are what we call history. Until cultural evolution began, the fate of human beings was decided by biology. For hunter-gatherers, it is decided by biology. If you want to hunt, you must be able to hunt. You must be able to capture prey. And if there are predators, you must run very fast, run away from them. So you must have the instincts of fight and flight. The cognitive revolution, according to Harari, is the point when history declared its independence from biology. And he has a sentence where he says, to understand the rise of Christianity, you can replace it by any other religion, or the French Revolution. It is not enough to comprehend the interaction of genes, hormones, and organisms. It is necessary to take into account the interaction of ideas, images, and fantasies as well. Cognitive neuroscience today is a very important field at the frontiers of neuroscience, and it in fact begins to overlap with what is being called today social neuroscience. But in Brunovsky's Ascent of Man, to which I must turn, Brunovsky has this wonderful sentence, and I must read this. He says, among the multitude of animals which scamper, fly, burrow, swim around us, man is the only one who is not locked into his environment. His imagination, his reason, his emotional subtlety and toughness make it possible for him not to accept the environment, but to change it. And that series of inventions, this is important, that series of inventions by which man from age to age has remade his environment is a different kind of evolution, not biological evolution, but cultural evolution. I call that brilliant sequence of cultural peaks the ascent of man. The invention of the tools of biotechnology contributes to evolution. The invention of the tools of information technology contributes to human evolution. Simply every invention of every kind, and remember, the invention of agriculture is what transformed human beings from hunter-gatherers to stationary societies. And cultural evolution began with the invention of agriculture. Cultural evolutionary theory, how culturally evolves and why it matters, is the subject of this paper in the Proceedings of the National Academy of Sciences, journal in which many of you would like to publish. Such papers do appear there. And what the authors say is that cultural transmission is more complex than genetic transmission and may occur on shorter timescales, even within a single generation. This is true because cultural transmission requires so many other factors in your environment. But can cultural evolution influence biological evolution? This is a very interesting question. It is addressed in this paper. Cultural traits can alter selection pressures and genetic traits and vice versa. And they say, and they show the example of lactase persistence. The enzyme lactase, which I show on the right side of the slide, breaks down lactose in milk to glucose and galactose. This is what is necessary for the digestion of milk. But for 100,000 years before the invention of dairy farming, human beings did not have milk except mother's milk from which they were weaned after infanthood. So there was, for much of human history, there was little reason to digest milk after weaning. Adults did not typically produce the enzyme that digests lactase because human genetic programs are such that very many enzymes responsible for the maintenance of the fetus and for the survival of the newborn get shut off as we mature. And this is one of them. Another one is the gene for fetal hemoglobin. The gene for fetal hemoglobin is shut off. And if you don't have a good gene for human hemoglobin, you end up having many other problems. However, with the cultural practice of cattle domestication and dairying, genetic mutation that enabled the production of the lactase enzyme in adulthood was strongly favored by selection. Because early human population which began to drink milk would have had diarrhea. Many of them would have succumbed because many techniques of hydration were not known then. And as a result, you would have a selective pressure. But is this understanding important? It is very important, as these authors point out. Today, if you had a job in one of the United Nations organizations which tries to improve nutrition in Africa, it would be wrong now to try and introduce dairy products into Africa, because as you look at this, the genetics, this has been done. If you have the brown, you have no lactase at all. If you have the red, then the lactase has persisted. So you can see lactase persistence is more in European populations. And if you come to India, lactase persistence is much more in North India than in South India. So many people, probably including many of us, get along because we've learned how to survive with the discomfort of drinking milk, because it is culturally important to drink milk. But I've come now to the very end of my presentation. I have on this slide the tree of life again, and tell you how important it is to understand what I call the unity of nature. But I wanted, when I was the director of the Institute, to invite Carl Lewis to the Institute when we were celebrating our centenary. But at that time, he wrote back saying that he was too old to travel. He died shortly thereafter. And then when I was searching the internet looking for an obituary on him, I found this wonderful tribute on the internet. And they had this picture or this image of Lewis. They had something that was said in Greek, which I did not understand. And then there was the tree of life. And they quoted Pliny the Elder, who apparently said, nature is to be found in her entirety, nowhere more than in her smallest creatures. I might just paraphrase this to say that chemistry is to be found in her entirety, nowhere more than in nature's smallest creatures, that is, in archaebacteria and in bacteria, in microbiology. Why do I draw your attention now in conclusion to such a disparate group of subjects? It is because of the environment in which we live, climatic, social, and political, in which we live today. Some reflection on the roles of chemistry and biology in shaping nature may allow us to introspect on the course of human history and attempt to understand why the world today is the way it is. As biochemists, we would like to understand why is biochemistry the way it is. Science is a deeply humbling subject. I hope I have conveyed to you how any attempt to understand even the smallest of nature's creations can be deeply humbling. In my very last slide, I would like to show you the coronavirus, which is a public health nightmare and an artist's delight. I draw your attention to a book by Peter Medawar, the famous immunologist and writer, called Aristotle to Zoos, which is a dictionary of biology. And if you go there and look under virus, you will find that a virus is described as a piece of bad news wrapped up in protein. That bad news is the nucleic acid which allows the virus to replicate. That's the bad news. And the protein is what covers it and allows it to enter our cells. It is that understanding that everybody is trying to get in the greatest molecular detail today. But you might ask, how long have coronaviruses been around? They've been around for a long time. When did we know about coronaviruses? We knew about coronaviruses only from the mid-1960s. And therefore, there's a lot to be learnt in nature, and there might be more viruses lurking around the corner. So in my very last slide, all I'd like to do is to acknowledge the two institutions where I have spent much of my very long career, which is now coming to an end. Much of it was spent at the Indian Institute of Science, and it is the undergraduates there who have really hosted this lecture. That's where I was there for everything, all the time until I retired in 2014. And the National Center for Biological Sciences here in Bangalore, which has now provided me a home over the last few years, and which is where I've really understood very much more the connections between chemistry and biology. Thank you very much.
Thank you, Professor, for that illuminating talk. We have some questions from the audience. So I'll just read them out to you. Unless you want to see them yourself, you can exit your presentation and...
I think I have exited my screen. I see all of you as letters, and I see myself at the bottom.
So at the top right of the screen, you should be able to see a Q and a question mark sign.
Question marks. Is it the chat box that I need to open? I've opened meeting chat.
Okay, just beside that should be the Q&A.
Show question mark. Show question and answers. Ah, wonderful. Yes.
Yes. So you have a list of questions over there.
Okay. Where do I have the questions? Open. It's opened. Published 11. What should I do now to see the questions?
If you click on published, I think you'll be able to see the questions.
I click on published. It says no new questions. Published is 12. They've come. Yes, yes. Do I start at the bottom or the top? Or?
I think you can start at the top because that's where most of the likes are concentrating.
Okay. I have no problem. Okay, please use this question block. Okay. Amazing. Most of the questions are anonymous. But I read the question, or people can read the question themselves. This is a question on vesicles are synthesized by extrusion of lipid solutions. However, in nature, a process requiring such extreme, so were the first protocells formed by random processes. Also, is it possible to computationally arrive at the time scale needed for these protocells to form? I'll first begin by saying I don't know the detailed answer to this question, but this is a question which has been around for a very long time. I think protocells can be formed spontaneously. In fact, it turns out that life itself is hugely improbable, which is why we haven't found it anywhere else so far, whereas the astrophysicists and astronomers and all have found all kinds of planets, galaxies, universes we don't know. But I think biochemically it's possible to form these spontaneously, yes. So in fact, there is a lot of early work where protocells were formed in primitive conditions, which is available in the literature. Yes, the probability of the formation of specific stable elements like carbon, nitrogen elements seems to be unlikely if left to chance itself. Yes, wonderful. That is actually the central question. Okay. I would only, thus here we begin to have the fundamental dilemma. Is there some force, some harmony of the universe at play behind these seemingly random events? Is there something along the lines of what people believe to be God? I would like to know about what biochemists like me think. It's not important to know what I think because I'm just an individual. And I might just say that I don't believe that there is any explanation for everything that we see other than the explanations that we've so far obtained from science. Science has gone far in getting very far in coming to where we are today. So I'm sure science in another 100 or 200 years would have gone much further from where we are today. So I don't think there is any fundamental dilemma here. But I'm not the only one who thinks so. This is a question which has been bothered because in the battle between the creationists and others, there's always been this question. So you will find that people like Richard Dawkins have written about this, but sometimes Richard Dawkins has written about it in a manner which is more likely to offend than to sort of mediate. But I think you should read a book by Jacques Monod written long time ago called Chance and Necessity. This is required reading for thinking about this question. And what we can do about this question is only think about it. Remember that when we invoke supernatural forces, we're just putting the problem elsewhere. What we are then saying is we don't want to think about it anymore. We've just accepted. The difference between religion and science is this. In religion, it is belief and you accept it. That's fair enough. In science, you don't accept anything. You should question what is accepted dogma today. And hopefully some advance will come out of this. Advances also come out of a lot of experiments that one does in the laboratory. But advances come also by just wondering. Go back and ask the question, from where did Jennifer Doudna get the idea for doing the first experiments on what has won her this year's Nobel Prize? You'll go back and see that this comes from work that nobody thought was ever important. I don't believe that work could have even been published in some of today's frontline journals. And that is simply looking at sequences in bacteria. So science learns all the time. The next question is, while chemistry serves as an important backbone in understanding biology and physics, can it be argued that biology and physics serve an important role in understanding chemistry as a subject itself? Yes, I would say, I may not phrase it exactly as you've done, not in understanding chemistry, but in allowing you to see new chemistry. That's what you would see in biology. Physics, of course, provides the fundamental understanding of chemistry. Without physics and understanding many things, which we take now for granted in chemistry. See, after all, understanding is something when enough people have understood it, there is no need for other people to understand it anymore. They just accept it. So a lot of science is also like religion, we just accept it. So every chemist does not now need to go back to quantum mechanics. They accept it. And I think that is what has happened over a period of a century or 2 centuries where science has progressed. Wonderful. I like the ones in which the questioner is not anonymous, only for one reason, because I find on the internet everybody is anonymous. As per the theory of spontaneous generation in biological evolution, 7 experiments have proved that origin must have started through the biogenesis. If this claim, how the first living organism's advent in this planet can explain, from the chemical point of view, how this unusual phenomenon can be. This is a question which you have not answered. The link has not been made between chemical and biological evolution. In fact, Lynn Margulis, the person who really gave us the idea that eukaryotic cells have really evolved by sort of engulfing bacterial cells, because that's the mitochondria. So you have a fusion between, say, an archaebacterium and another bacterium, which now to get the origin of organelles in cells. But where did the first cell come from the chemical point of view, nobody knows. This is still something that I have a feeling is going to go on for a long time. We don't know. Lynn Margulis in a famous article says this, that you can't take a mixture of chemicals and just add them together and get a cell. This is not going to happen that way. So how did it happen the first time is something that we don't know. In fact, for this reason only, the famous astrophysicist Fred Hoyle thought that introduced the idea of panspermia. That is, life on Earth was seeded from outer space. But that was only pushing the problem elsewhere. And he had this wonderful correlation between sunspots and viral epidemics. So even at the beginning of the coronavirus pandemic, somebody wrote to me asking, is there a connection between sunspots and this? And then I told them about Fred Hoyle. Fred Hoyle would have won the Nobel Prize for stellar evolution of the elements in the stars if it hadn't been for the fact that he espoused panspermia. But we still don't know. That's all I can say. The next question is on SARS-CoV-2. What is the specialty in them that is making them deadly? Why is the SARS-CoV-2 so different? See, I don't think the SARS-CoV-2 is awfully more, I don't know, more terrible than SARS-CoV-1, which appeared in 2003. SARS-CoV-1 was controlled very quickly, and it also came and was the first cases were found in Vietnam, came from southern China. It was controlled very quickly at that time. This time, because there's been much more travel and recognition came slowly, it has spread. I don't know whether the SARS-CoV-2 virus is more deadly than SARS-CoV-1. I haven't seen laboratory things which say this. But how did it come about? There's still discussion. Nobody knows here. The next question, the cosmogenic theory of Richter. This question, I'll definitely get the answer. I don't know. So I will move. Questions which I don't know, I shouldn't say anything. And question again on the importance approach in studying characteristics, how they may interact. However, in many systems, especially the human brain, the question of consciousness is being hypothesized. Breaking down may not be sufficient. So we have the whole greater than the sum. How can... No, I think this whole greater than the sum of the parts is only a way of somehow getting away from the painstaking work that is needed to construct the whole from the parts. All the painstaking work of biochemistry was done in the early part of the 20th century. And we've come this far. I've given you one example in this lecture itself, the example of Anandamite. That controls your emotions. It's one molecule which is acting on one receptor. And just think about marijuana. Most of the people in India have been using marijuana for a very long time. And even today, marijuana is very important in contemporary discourse because the National Narcotics Bureau does not know whether 150 grams is important or 1.5 kilograms is important or 1,000 kilograms is important. So so many things. All the hallucinogenic molecules are known today as to which receptors they act on. So we will have a much better understanding of neurochemistry as techniques improve. The enormous improvement in chemical techniques, mass spectrometry imaging, mass spectrometric imaging, NMR imaging, all of this. So I think science is producing techniques which we never had. So I think people who like to theorize always like to say that we are not going to get a detailed understanding. Next question is cultural evolution like natural selection, like humans who cannot digest would be on a significant disadvantage when compared to other people. I don't think so, as long as they control dietary intake and have appropriate diets. People have lived quite well without it. Many people, even in India, who don't
who have lactose intolerance have recognized it, who live quite well. How many more are there? Yes. Can we term the origin of life an accident? Undoubtedly, isn't it? We are all here because of a happy accident. As Brunovsky says, even the collision of 3 helium is a fortunate accident. And yes, we are here by accident. We'd like to think we are here by design. But one thing an understanding of design of evolution teaches you is evolution probably has no purpose. And it's very difficult to imagine a life without a purpose. Optimization of enzymes and changing are already well. How in recent revealed the changes in optimization may lead to architectural changes. This is evolution in the test tube. This is what people are doing, for what Francis Arnold got the Nobel Prize a couple of years ago, for trying to get bacteria to do new things which they hadn't done before. And I think in this more progress is going to be made. I think we haven't heard the last word on this so far. I think that's all the questions. Oh, which I can see at least, I hope. Oh.
I think I can see a couple more questions.
Can you tell me what they are?
Sure. So the last question you spoke
below
about was...
your subscribe to our newsletter.
Yes, yes.
I didn't scroll down. I forget this all the time. Yeah. How did agriculture help in evolution in humans? I'm afraid even to tell you the answer, because this is there in Harari's book, and I like his explanation very much. See, once you invented agriculture, human populations had to become stationary. They had to become stationary to look after the field, and also to protect it from animals which would come and eat up whatever you were trying to grow. And over time, if other people also felt that what you were growing was useful to them, they would come, other tribes would come there. This required the formation of a society. Or early societies were only tribes. In fact, even modern societies are a collection of tribes. You know, our worst instincts are tribal instincts, where we join into groups and protect from other groups. So agriculture is the human invention. It's a human invention. It's a scientific invention. Agriculture is a science. And that invention transformed human cultural evolution. But as your societies grew a bit larger, how do you keep them together? You need myths. You need to create some stories. You need to create something which will bind people together. Those binding myths are the myths which bind all of us together today. That is religion. Harari was dissuaded from using the word religion by his publishers so that his book would sell better. But that's what he really meant. And I don't believe he's the first person to say it, but I believe he's the first person to articulate it in a readable fashion so the rest of the people can understand what this idea really is. You can agree with it or you can disagree with it. How can we be so sure that in other planets are different and evolution will result in a similar? Why am I so sure? Because if I look at Mendeleev's periodic table, and all of you who hate chemistry, it is almost impossible to imagine chemistry now coming from another set of elements which is going to be as diverse, as productive, because you need chemical diversity in order now to fuel biological diversity. And some kinds of elements, your lecture series is entitled Elementary, and elements are anything but elementary. And only with a certain set of elements can you have all of organic chemistry. And if you go to the inorganic chemistry department, they will say, we like all the elements which are not a part of organic chemistry. But if you look at all of these in totality, you will see this is still only a small part of what is there in the Earth's crust. Can chemistry or biology have an impact on physics and computer science? This is, I don't know whether you're asking this because you know something or you're a young student. If you're asking this because you know something, then you should stay anonymous, because my answer would be to rephrase your question. I can say I want to ask, how can chemistry or biology have an impact on physicists and computer scientists? Because finally it is people who change fields. And it turns out that biology has had more of an impact on physicists and computer scientists than any other discipline. Computer scientists have had genetic algorithms for a long time. Physicists have migrated at different points in history from physics to biology. Delbruck, who was Niels Bohr's student, led the first lot. Walter Gilbert was a physicist, worked with Abdus Salam, who developed the chemical method of DNA sequencing, for which he got the Nobel Prize. You don't use it now. Sanger's method is used. Sanger's also a chemist, a trained chemist, of course, invented a different method, more convenient. Computer scientists, physicists who work in statistical physics, look for examples in biology. And in the IISc itself, you will find physicists in ecology and all. So computer science, I think, is what everybody needs. And computer scientists need a rationale. And the rationale they find in biological information. Today, when you talk about data science, what data are you going to handle? You're going to handle biological data. That's the large data, whether it's medical data or genomic data. Just like carbon, silicon can also bind for molecules. So is there a chance for atoms? It should be. So is there a chance that in outer space, there's a chance that life is made up by silicon instead of carbon? Now you should ask, I'll ask you a counter question. Silicon is hugely abundant on the Earth's surface. All sand is silicon dioxide. Why then is silicon not used in organic chemistry? You can ask this question. The only place I know where silicon, and people have studied a little bit, is silicon is uptaken by some strains of rice. And there is a little bit of uptake. Silicon is very highly oxidizable and eventually goes to silicon dioxide, which is sand. And this is why. See, oxidation and reduction are things which everybody hates in chemistry. And all of chemistry is oxidation and reduction. Because, you know, if you talk in terms of a physicist, take an electron, go there. You take away an electron, it becomes oxidation. You add an electron, it becomes reduction. So every process is like this. Keeping in light chemical evolution, viruses seem to be chemically simpler. Viruses were also coexisting before the first living. That's a wonderful question. In fact, I think it'd be important, you know, that there were crystalline viruses there on Earth as the first things which formed, but they couldn't replicate. And then bacteria came along and you get bacterial viruses. It's a wonderful thought. One should think about it. I don't know whether there's any experiment. Trait unit. What biochemical curve? No, biochemistry, this is a wrong question. A biochemistry and what can I say? I better use not the mouse with this. I missed a question. What I have used about biomimicry. Biomimicry is wonderful. You'll find it in biology all the time. And but that mimicry is somewhat more sophisticated mimicry. It's not chemical mimicry sometimes. It's a way in which organisms learn how to camouflage themselves, look like other organisms, avoid predators, all kinds of things. Keeping in light chemical evolution, viruses seem to be chemically simpler. They might just have been. I do not know the answer. come to the bottom. Is sapiens trait unique? What No, all biochemical processes in animals would be the same. You can only ask what is the genetic component that is now being expressed. See, between chimpanzees and us, I don't know, we are some 99 point plus percent the same. It's a sobering thought. And now nobody knows what, and this is again, the human genome and the chimpanzee genome have been known for 20 years. We can't answer that question. This is why you should keep studying so you're able to answer that question at some time or the other. Remember that in science, that every question you ask is already answered. Then you must ask the more important question. What am I doing? Because there's no more question to answer. That would be the end of science. Not going to happen for a while. About the watchmaker's theory, cannot we have a survivor fallacy there? Because we have so many combinations, we turn out to be the conscious ones because other combinations fail. Quite possible that there have been many, many experiments which have been failed. And even on Earth, there would have been so many experiments which failed, so many extinctions which happened for one reason or the other, so many organisms which had short-lived spans disappeared. Dinosaurs looked fearsome, but they disappeared. And we are still debating why they went extinct. And yes, it is possible other combinations have failed. But that's not the survivor fallacy. It just says that as survivors, we haven't yet found other survivors. If we found other survivors and they were different in some ways, we'd have something to study. See, the trouble with, I think, what I call the millennials and Gen Z, which is where all of you belong. The millennials, who are the ones who came after 1980, I think, or thereabouts, and the Gen Zs are those who came after 2000. And is that you haven't grown up with science fiction. And those of us who grew up with science fiction, even when we are about to leave, we still hope some wonderful creatures will be found somewhere or the other. Because those were the books that we read, those were the comics that we read, those were the movies that we saw. And occasionally we may still see those movies if they are replayed on TV. My wife still asks me, why do you watch Jurassic Park for 100th time when you see it? But I had a dinosaur fascination, and I guess most people had at that time, and you still worry about it. Yeah.
Thank you, Professor, for taking all of these questions. We had quite a lot of questions today. Unless you have anything else to add, I would switch over to Nikita to convey the vote of thanks.
No, I don't have anything to say. I can't hear you. What happened?
Hi, can you hear me now?
Yeah, I can hear.
Yeah, sorry about that. Thank you so much. A lot of people came to attend the talk, and I think they were really, really enthusiastic, and hence like the extreme number of questions. Thank you for being so patient. So yeah, firstly, we would like to extend our heartfelt gratitude to Professor P. Balaram for accepting our invitation and taking time out of his day to give us this extremely fascinating talk about chemistry, biology, and nature. I think this talk beautifully highlighted the intrinsic relation between chemistry and biology and even gave examples of this relation in various domains of research. We would like to thank the Dean of our UG program, Professor P.S. Anil Kumar, for making this talk possible. We would also like to thank the rest of Team Pravega for supporting us and helping us in the organization of the talk. And lastly, we would like to thank all of you for attending this talk, and we hope you enjoyed it. If you did, Pravega is hosting a bunch of other talks which you can check out at our YouTube channel. This talk will also be uploaded on our YouTube channel in a few days. The next talk of the elementary lecture series is COVID-19 and probing pharmacology and biophysics of viral proteins, which will be given by Professor Michael Marty from the University of Arizona on 14 November, 7:30 p.m. Till then, goodbye and take care.
Okay, bye.
Right. With